This animation deals with mitosis, or cell division. Here on the screen we have a representation of the nucleus of the cell. Normally within humans we have 46 chromosomes, which consist of 23 pairs. One member of each pair inherited from your mother, the other member of each pair inherited from your father. For ease of representation, I am only showing 4 chromosomes in this animation. 2 from your mother, 2 from your father. They're represented as a green pair, where the centromeres are shown in green, and a red pair, where the centromeres are shown in red. One member of the green pair inherited from one parent, the other from the other parent, and same with the red pair of chromosomes. So let's have a look at how a cell divides into 2 daughter cells. Mitosis is a process which makes identical cells. So initially we have a prophase. Prophase is where the chromosomes condense. Initially they start looking like a big mass of spaghetti. You can't define the individual chromosomes. But during prophase they all condense so they're in neat structures to be able to be separated into the new daughter cells. After they have condensed in prophase, they then move into metaphase. Meta stands for middle, and in this phase the chromosomes line up along the midline of the cell. This is in preparation for division. It's where the spindle fibres would start to attach to the centromeres, and the chromosomes are going to be pulled in opposite directions. Each chromosome at this point has 2 sister chromatids, because it has replicated its DNA during the S phase of the cell cycle. During the next phase of mitosis, which is anaphase, is where we are going to separate those sister chromatids so that one chromatid from each chromosome will be pulled to one of the poles of the cell. Just like the Earth that has a North Pole and a South Pole, a cell has 2 opposing poles. So let's watch what happens during anaphase. Those centromeres split and one member of the chromatid from the original chromosome will move to one pole and the other chromatid to the other pole. As soon as that centromere splits, they're no longer called chromatids. They are now chromosomes in their own right. Then we go through a telophase, and the telophase is where you pinch off this one cell into 2 new daughter cells, with each cell now containing 4 chromosomes, as did the original cell before it divided. The difference here now is that DNA has not yet replicated, so you don't have chromosomes with sister chromatids, you just have individual chromosomes. For simplification in this animation, I haven't shown all the nuclear structures or the cell structures. I've just been showing what's happened to the chromosomes. So, we started with one cell with 4 chromosomes, and we now ended up with 2 cells, each with 4 chromosomes. In humans, we would have started with a cell with 46 chromosomes, and we would have produced 2 identical daughter cells, each with 46 chromosomes.